Good morning, good afternoon, good evening, ma'am. So, we present the Germline Gene Therapy. Germline Gene Therapy targets the reproductive cells, meaning any changes made to the DNA will be passed on to the next generation. Consequently, the practice has dramatically divided opinion. Germline Gene Therapy, also called inheritable genetic modification or IgM, means changing the genes in egg or sperm cells or in the cells of very early embryos, and that alters the reproductive cells of a person's body. And now, we provide a short video presentation shows germline gene therapy is capable in preventing diseases to be passed from mother to their children.
A mother's egg cell contains thousands of mitochondria, each containing its own DNA. If a defect, the DNA in these cells will pass the diseases from mother to the child. In germline gene therapy, the nucleus is removed from the mother's egg cell. The nucleus is also removed from an egg cell provided by a donor. The nucleus removed from the mother egg cell is inserted into the donor egg cell. Thus, the donor's normal mitochondria replace the mother's defective mitochondria containing mutated DNA. A sperm cell is injected to fertilize the egg. The cell is then reimplanted into the mother and develops into a healthy baby.
Gene therapy ethical. German gene therapy is when DNA is transferred into the cells that produce reproductive cells, egg or sperm, in the body. This type of therapy allows for the correction of disease-causing gene variants that are certain to be passed down from generation to generation. However, germline gene therapy is currently not legal in the US because the risks still appear to outweigh the benefits. Next, somatic gene therapy. Somatic gene therapy is when DNA is transferred into body tissues. It specifically targets cells in the body which are not passed on to the person's children. As a consequence, it doesn't raise the same ethical issues as germline gene therapy.
Gene therapy medicine is at a turning point on the cusp of major change as disruptive technologies such as gene, RNA, and cell therapies enable scientists to approach diseases in a new way. This swiftness of this change is being driven by innovations such as CRISPR gene editing, which makes it possible to correct errors in DNA with relative ease. Process in this field has been so rapid that the dialogue around potential ethical and safety issues is scrambling to catch up. This, di- this disconnect was brought into stark relief at the second International Summit on Human Genome Editing, held in Hong Kong in November, when exciting updates about emerging therapies were eclipsed by the disturbing announcement. Caingcoy, a Chinese researcher, claimed that he had edited the genes of two human embryos and that they had been brought to term.
In Germline Gene Therapy, there was an immediate outcry from different scientists across the world, and he was subjected to intense social pressure, including the removal of his affiliations for having some allegedly disregarded ethical norms and his patients. And as Glenn Cohen, a faculty director of the Fit Reforms Center for the Health Law Policy, Biotechnology, Bioethics at Harvard Law School, said that the gene editing comes in many different varieties with many consequences. In any deep ethical discussions, needs to take into account those distinctions and the use of CRISPR technology to be used in editing of genes and to change around the world.
So, somatic gene editing compared germline gene editing. Germline editing carried out is quite different from the somatic gene therapies that are currently changing the frontiers of medicine. While somatic gene editing affects only the patient being treated and only some of his or her cells, germline editing affects all cells in the organisms, including eggs and sperm, and so is passed on to future generations. The possible consequences of that are difficult to predict. Somatic gene therapies involve modifying a patient's DNA to treat or cure a disease caused by a genetic mutation. Germline human genome editing alters the genome of the human embryo at its earliest stage. They may affect every cell. So, this means in one clinical trial, for example, scientists take blood stem cells from a patient, use CRISPR techniques to correct the genetic mutation, causing them to produce defective blood cells, then infuse the correct cells back into the patient. The treatment changes the patient's blood cells, but not his or her sperm or eggs.
Gene therapy controversies and treatment approaches. So, in employing new medical technologies like gene therapy, stem cells and pharmacogenomics are good for our own. So, in vitro fertilization or IVF have a miracle in the late 1970s. So, the miracle is it is occurred 5 years before that the polymerase chain reaction in the field of genetics. Second is the 18 years before the first bacterial genome was sequenced. Third is the 20 years before completion of the human genome sequence. And lastly is the 30 years explosion knowledge of mammalian biology. So throughout this period, many members of the general public, especially those incurable disease or loved ones. Gene therapy is a single nucleotide polymorphism or SNPs. A copy number variants or CNVs, it is not harmful, but it is focuses on correcting the random insertion of the normal gene into the genome. It is the most common technique and focus in replacement of the abnormal gene with the normal one and repair of the abnormal gene. Last is altering regulation of a particular gene. Ethics of gene therapy. The ethics of gene therapy is to cure the fatal genetic disease differs from more traditional remedies like surgery, pharmaceutical, and physical therapy. And it is frightening to some people. So, like what happened to Gaucher's disease, a lysosomal storage disorder, a phenylketonuria or phenylalanine hydroxylase deficiency, doxin muscular dystrophy or DMD. Scientific obstacle. A gene therapy are related to the vehicle used to deliver normal gene. This virus carries the gene into the patient's body and to the targeted tissue. Stem cell therapy. Beyond gene therapy, another issue of much debate relates to the use of stem cells. So these cells can be divided into two broads. First is the embryonic and second is the adult. But both classes are currently being explored for possible therapeutic application. What is embryonic stem cells? So embryonic stem cells is a chromatin from donor cells joined to form pronuclei in the oocyte cytoplasm. And what is adult stem cells? So adult stem cells is all tissues and organs contain undifferentiated cells. Next is induced pluripotent stem cells. So according to Takahashi et al. 2007 and Yu et al. 2007 stated that induced pluripotent stem or IP cells that rival an ES or the embryonic stem cells in their ability to differentiate have been generated using adult human somatic cells.
Ethical controversies involving stem cells. Treatments using adult stem cells rarely generate controversies. As a matter of fact, bone marrow transplants have become so familiar as a cancer treatment, that many people may not fully grasp the concept that stem cells are being used. Pharmacogenomics, the study of the role of the genome in drug response. It reflects its combining of pharmacology and genomics. It's analyze how the genetic makes up of an individual affects their response to drugs. And also, pharmacogenomics has recently taken on new importance of three reasons, and that three reasons stated by Phillips and Van Bieber 2005.
Pharmacogenomics has recently taken new importance for three reasons. First is new tests are being made available, broad use rather for and the narrowly target population. The second is concern about their safety of drugs such as Vioxx and Celebrex will likely lead the FDA to use pharmacogenomics testing to a great extent to increase drug safety. Number three is so far has been little support early assertion that pharmacogenomics will reduce healthcare costs and adverse drug reaction.